Hello everyone, welcome to Chandu Biology classes, and today is the lecture 3 of our series for CUET PG and GAT-B, in which we are covering the most important topics for your upcoming examination. In this lecture we will be dealing up with Photosystem 2. So in the previous lecture I taught you about the structure of Photosystem 1, and I went into the great details of what are the different subunits of the photosystem 1 and what are the inhibitors of photosystem 1, and we solved somewhat questions on the same concept. Now in this lecture we will be dealing with photosystem 2 and oxygen evolving complex also, how oxygen is being evolved and how splitting of water happens, what are the key residues which are important. Okay, and we will also answer that question that I asked you in the previous lecture, that the damaging of D1 protein, how it is helpful in photooxidation. That is also something that I will be answering. And the first question that I asked in the first lecture, that which is the correct equation of photosynthesis, for that you have to wait for our upcoming lectures. Okay, so my name is Deepesh Joshi and I am an third year integrated PhD scholar at ISC Bangalore, and I have qualified all these examinations. Basically, today my teaching is for these series. For GAT-B and CUET PG, we are focusing on these two examinations. Okay, and we are covering all the important topics. So just you have to follow the lectures, solve the questions with us by pausing the video, and practice on your own. Revise whatever is being taught. The content is more than sufficient, so don't need to worry. Okay, so let's start with the lecture. Okay, so basically, I told you that there are different kinds of arrangements of the thylakoid in the chloroplast. So the thylakoid, it could be something that is more exposed to the stromal side. If it is more exposed to the stromal side, then it is called as non-oppressed. Okay, that means it is more accessible to the stroma. Correct. And the ones which are sandwiched by the neighboring thylakoids, those are called as oppressed thylakoids. Okay, or non-stromal. Okay, and these were stromal and non-oppressed. So this was the classification of the thylakoids based on the accessibility to the stroma. So in this diagram, you can see that there is a distinguishable pattern of arrangement of these different molecules, where you can have the LHC1 dimer, LHC2 trimer. I told you LHC1, I told you a trick to remember. After 1 in the number line you get 2, so LHC1 will be dimer. After 2 in the number line you get 3, so let's see 2 will be a trimer. Okay, so basically here you are having the LHC2 complexes, then this is your PS2, and you can see that you also have your LS1. Okay, and this is the LS1. Okay, and you are having different sorts of, so this is your PS1. I told you PS1 photosystem 1 is associated with LS1. Okay, the and another thing to note is that this blue one, cyan one, this one is your cytochrome B6F dimer. Okay, so this molecule is also dimeric. And then this is your ATP synthase, that is something to synthesize ATP. ATP synthase, from the name itself you can guess the function. Okay, so this is the typical arrangement of the oppressed, non-oppressed thylakoids and the components which are present. Okay, now moving over to the next thing, we can see that what is the percentage of thylakoids. So basically, if you see the oppressed thylakoids, okay, and what are the components. Okay, so basically you can see that you can have oppressed or stromal exposed, and these are the different percentage of the components which are present. In the stromal exposed thylakoids, primarily you are having the PS1, that is P700. In your PS2, that is P680, that is primarily present in the upperest thylakoids. Cytochrome B6F complex, which is dimeric complex, that is present in the ratio of 1:1, 50:50. Okay, this is very, very important. Then LHC2, that is present 90% in the upperest thylakoids, 10% in the stromal exposed thylakoids. Okay, the ATP synthase that we study, basically that is not present in the upperest thylakoid, that is 100% present in the stromal exposed thylakoids. Very, very important. Plastocyanin, that is 40:60 ratio. So that is not much important, but ATP synthase, your LHC2, cytochrome B6F, and PS1, PS2, these things you have to remember for your examination. Okay, now once we are crystal clear with this concept, we all know that the light harvesting complex that is present. So basically in this thylakoid, you are having these units which are called as photosynthetic units, also known as quantasomes. And these quantasomes, they consist of these reaction centers. The reaction center is accompanied by the pigment molecules which are called accessory pigments, antenna molecules, light harvesting complex. Okay, all these can be the name of these accessory pigments. So if this much concept is clear to you everyone, I will just ask you a question, that thylakoid membrane has lateral asymmetrical positioning. Very, very important point in the question itself. Asymmetrical positioning of photosystem. We have seen that diagram there, everything was asymmetrical. Nothing was in proper symmetry. So, which statement is correct for the distribution of PS1 and PS2? So now, in this question, as you can see, that first thing, we notice that there are two kinds of thylakoids, and in the table only I told you that primarily PS1, it is present in the no oppressed or non-oppressed portion, and PS2 is present in the oppressed portion, that is the one that is sandwiched. This is the oppressed, this is the non-oppressed. Okay, and the stroma lamellae, the thylakoids which are there, these one are also non-oppressed. Okay, so here you always have higher abundance of PS1, and the oppressed you have the abundance of PS2. So basically first is your correct answer. So that is how you answer the questions. Now the major difference in PS1 and PS2 found in chloroplast are. So you have to tell, like, what is the major difference that comes in your mind till now, whatever I have taught you, what is the major difference? So basically the major difference, is it in the position of grana? Is it in chlorophyll A? No, chlorophyll A is common to both of them. Is it in the position of the electron carriers? Okay, is there a difference in the position of the electron carriers, or in the light harvesting complex? So the light harvesting complex is the same, position of electron carriers remains the same, only difference is in the position in the grana, that the PS1, they are more abundant in the non oppressed part, that is the stromal thylakoids, and PS2 is more abundant in the oppressed part. Okay, so this is something that is to be considered and to be remembered by you. Now since friends, now you are able to understand this much, let's deal into the structure of photosystem 2, okay, in detail. Okay, so let's see how it happens, like what are the different components. So basically if you focus, let me focus only on the structure for now. So basically you can see I have told you that the LHC2, okay, LHC2 is a trimer, right? So you can see that the photosystem 2, this is the whole PS2, this PS2 is associated with the light harvesting complex 2. Okay, and this is a trimeric molecule, I also told you what is the overall molecular weight of the trimer, 75 kilodaltons in the previous lecture, guys, lecture 2. Just follow all the lectures in the sequence, because if you are attending directly this lecture, then previous part will not be clear to you. So just attend in a sequence. So the overall molecular weight of the complex was 75 kilodaltons, and for each trimeric molecule that weight will be 25 kilodaltons. Okay, that is 75 by 3. Now, so here in the PS2 you are having these different proteins, which is CP43, CP47, D1, D2. Okay, these are different proteins, and there is a MSP protein also, which is nothing but manganese stabilizing protein. Okay, that is also associated with the PS2 only, that is the P680. And this is something that is important for the oxygen evolving complex. For that particular part, this MSP plays a crucial role. What is the role, what are the residue involved, and all those details we will cover in the upcoming slides. So for now you need to know that there are different electron carriers, okay, which are present. Basically you are having this PQA, PQB, you have this pheophytin. Okay, once light is incident, the flow of electrons happens, okay, from pheophytin to PQA, PQB. Okay, PQ is plastoquinone, and similarly like this flow happens, and then the electrons are further transmitted. That flow we will consider, and we will study that in cyclic and non-cyclic photophosphorylation. But for now you need to be very much clear with the different components. You have the CP43, CP47, D1, D2 proteins, and MSP. LHC2 is associated with photosystem 2, that is P680, which absorbs at 680 nanometer. The reaction center basically absorbs at this wavelength. Okay, so this is something that you should know. Now there are different genes which code for this protein. Now if anyone remembers who has attended the lecture 2, that for PS1 all the genes which were present, whether it is LHC, okay, something, so that starts with this A, okay, PSA. Okay, so here after PS you have a. So whenever you are having this A written in small, that gene will correspond to PS1. And whenever you will have the genes with B that is being written after PS, okay, or after any sort of initial prefix, then if B is written, then that corresponds to PS2. So this is a very simple trick to memorize the genes, because many people, they are like, they don't remember, they can't recall at the examination time that which gene is coding for proteins corresponding to which photosystem. So for that I have made the trick that if something is written with small a, okay, considering the prefix, then that particular gene is coding for proteins for PS1, and if it is with B, then it is coding for PS2. Now, the PSBA, it codes for the D1 protein. Okay. The PSB D gene, it codes for D2 protein. PSB B gene, it codes for CP47, and PSBC gene that codes for CP43. So this is something that is, these are the genes which are coding for these proteins. And if you consider all these genes, guys, all these genes, basically they are encoded by the chloroplast genome. There is nothing to be do with nuclear genome. But there is another gene that is PSBO. Again, you can see here is a B. That means for PS2. So this codes for oxygen evolving complex, or MSP, that is manganese stabilizing protein. Okay, so that is basically this gene. It is coded by the nuclear genome. Okay, it is coded by the nuclear genome. So on the thylakoid membrane, the stromal side, the lumen side, On the lumen side you have this MsP, and on the stromal side you have the CP43, CP47, D1, D2 proteins. Okay, and the flow of electrons, they happens from this pheophytin to PQA to PQB and further to more carriers that we will see in the upcoming lectures. So this is a brief in a nutshell about photosystem 2. Okay, now if this part is clear to you, we can move on to the inhibitors of this photosystem 2. So basically the inhibitors mainly they target the electron flow or the electron movement from the PQA to PQB. So this part is being targeted. So since this part is targeted, how it is targeted, guys? So what happens is, let's say this is your PQB, okay, and this is your PQA. So what happens is that once this PQA receives the electron from pheophytin, then this has to transfer the electron to PQB, but the site where the residues where it has to accept the electron, at that particular position these inhibitors, okay, this DCMU or this inhibitors, this sits at this particular position and they block the electron flow. Okay, because if this is sitting over here, it can't accept the electron. So that is how this is blocking the electron flow, and this is inhibiting PS2. Okay, because these components are present in PS2. Okay, yeah. So these inhibitors, they can be urea derivatives. If they are urea derivatives, they can be CMU or DCMU. Okay, so the CMU, these are monourons, that is chlorophenyl dimethyl urea. This is the name of the inhibitor. This is a monouron, and it can be a diuron like DCMU. So the DCMU, this is very important from all PYQs point of view, because in all PYQs there are questions on DCMU, and this DCMU basically it inhibits PS2. This is something you need to remember. Within that PS2 specifically, what it inhibits your electron transition from PQA to PQB. Okay, then you can have triazine derivatives like atrazine, simazine, which works in the similar fashion. And you can also have uracil derivatives like bromouracil, that is important, and isosyl. So all these are the inhibitors. Okay, so if you consider this in more detail, I have drawn the structure of DCMU. Like here is the structure I have attached. So DCMU is dichlorophenyl dimethyl urea, that is a diuron. This is the structure, two chlorine groups, okay, dichlorophenyl, okay, dimethyl urea. Okay, so this is the structure. DCMU action is it block electron flow by occupying the binding site of plastoquinone competitively where normally QB binds, that is plastoquinone B binds. Okay, so that particular site, what it does, it occupies the site and it competitively inhibits the binding of QB. So if QB can't bind, okay, what will happen, it will not be able to accept the electron that is being transferred. This is what is happening. And the triazine, it block electron flow by occupying the binding site of plastoquinone. Again, similar mechanism of action. Paraquat, I told you, it inhibits PS1. Okay, it inhibits PS1, that I told you in the previous lecture. So here, if this is the photosystem, this is the PS2, this is the PS1. Electrons, basically if light is bombarding, electrons are flowing. Once P680, it absorbs the electron, the wavelength corresponding to 680 nanometers, it gets excited, it reaches an excited state. In the excited state, it transfers the electrons, it comes back to ground state, it transfers the electrons to the plastoquinone A, then it should go to plastoquinone B, but in the process, in the transmission only, this DCMU is causing the inhibition. Okay, and paraquat, as I told you, that this paraquat, it like finally accepts the electron and doesn't let any d plus to get the electron. Okay, so that is why this paraquat inhibits the PS1. Okay, that is how it inhibits. So basically this is about the inhibitors. Okay, and this is the overall summary. This is the summary. Okay, very important summary. Okay, here I have summarized all the inhibitors which are important. Okay, so you have this DCMU which is inhibiting, this is the PS2. This DCMU is inhibiting the electron transition or the electron flow from the QA, okay, that is the plastoquinone A to plastoquinone B, okay, by sitting at the site of QB, okay, and inhibiting the electron flow. Then DBMIB, this is something I told you in the last class. This inhibitor, it basically inhibits cytochrome B6F. Okay, so it blocks the electron flow from the PQ to the cytochrome B6F. And then you are having plastocyanin P700, FX, FAB. So I told you all these iron clusters, the genes which are responsible, okay, coding for the proteins, all those things I have told you, PSAC and everything. Then this paraquat, it inhibits basically, it stops the electron flow to NAD plus, and itself it accepts the final electron, and it leads to superoxide formation. These superoxides, they can damage the lipid component of the thylakoid, and it can lead to damage to the plant. So basically these are the all inhibitors. One is DCMU which inhibits PS2, one is DBMIB which inhibits cytochrome B6F, third one is paraquat that inhibits your PS1, that inhibits your PS1. Okay, so this is something you need to remember, guys. And this is the detailed flow of electron that is happening. Okay, so basically here you can see that if I zoom it in, okay, here you can see that this is the overall diagram, this is the stroma, this is the lumen, and this is your overall PS2. So in the PS2 you can see as soon as the light is striking the reaction center P680, which consists of chlorophyll A, it is moving to excited state, coming back to ground state, releasing an electron. Electron is passing through this pheophytin to the QA, that is the quinone. Okay, and when it passes through the quinone, then it again passes from QA to QB, and once the electron is passing through QA to QB, then there is another sorts of carriers which are present. Okay, so basically here you are having this MSP, that is the manganese, okay, supporting protein, that is the part of the oxygen evolving complex also, OEC, okay, or the water splitting complex also. So this is the part of that. So here you are having this 4 manganese atoms, and because the P680, once it has like lost an electron, it becomes deficient in electron, and this will be taking the electron from this Z. Now what is this Z? What is this Z, guys? So this I will tell you in the further slide. What is this Z? Okay, so you can see that here there is the reduction potential that is being plotted over here. So the flow always happens from the region of high reduction potential to lower reduction potential. What is reduction potential? It is the tendency to accept electrons. It is the tendency to get reduced. That is what is we call as a reduction potential. And then similar thing is happening in this diagram also. The electron is flowing there. Here you have the time that is being shown over here, that is in microseconds this transmission is taking place. This happens like really in a short interval of time, okay, all these transfers. So basically this QA, that is the quinone, is capable only of single electron reduction to semi-quinone. Okay, so this is tightly bound to the photosystem. The QB, so electron flow happens like this, QA to QB. Okay, so the QB can switch between 3 states. it can be fully oxidized quinone, okay, it can be semi-quinone, and it can be fully reduced quinol. So basically, if it is in this state, it is called as fully oxidized. If it is semi-quinone, it is represented by this, and if it is fully reduced, then it is represented by this symbol. So it is loosely bound, okay, and it is present in the hydrophobic core of the thylakoid membrane. So this is something which is important about the QA and the QB. Okay, so QA is capable only of a single electron reduction to semiquinone, whereas QB can switch between the fully oxidized QB stage, quinone, then semiquinone, and at last fully reduced. So quinone to quinone, it is getting converted, it is reducing to quinol. Quinol is what? Alcohol. On is what? Ketone. So there is a reduction that is happening by accepting electrons. Okay. So there will be an intermediate transition state that will be called as semiquinone, QB negative. And this one will be the fully reduced form that will be represented by this. Okay. And this quinone state, that is fully oxidized state, that will be represented by this. So this is the overall picture of the quinone and the electron transfer that is happening in the photosystem 2. Now if you see this, we are talking about the cytochrome B6F complex, guys. So here this is the stroma, this is the lumen. As soon as the light corresponding to 680 nanometers or lesser, okay, lambda that is less than equal to 680 nanometers is falling on PS2, what is happening? It is transferring the electron, okay, the electrons are getting transferred, and as soon as electrons are getting transferred, what is happening is that mainly the first thing is that when you are passing the electrons, okay, then as you can see over here, that you pass the electrons like to pheophytin, then to QA, that is quinone A, then to quinone B, and then to plastoquinone. So when you are passing the electron to plastoquinone, what is happening? This plastoquinone, it is first getting reduced to PQH2, and this PQH2 then again it gets oxidized, and as soon as it is oxidizing, it is like transferring the electron. Okay, the electron is being transferred through the different sorts of proteins which are present. So the cytochrome B6F, it is a multi subunit complex protein, plus it consists of many prosthetic groups. So basically they are the cytochrome B, cytochrome F, then you have the heme B, heme C, and the iron sulfur proteins. So basically the flow of electron is happening like this, okay, and since the electrons are transferred from this all these proteins, basically you are having the iron sulfur complex that is called the risque protein. In the risque protein, this is a 33 kilodalton protein. Here the electrons, when they pass through the heme B or your heme C and all this, then it is transferred to the risque protein and then to the cytochrome F, where you are having the heme C, okay, and then this is plastocyanin. So then plastocyanin gets the electron, okay. So basically this is the overall flow that is happening, and in this entire process into the lumen, you are pumping in 2 protons. Very, very important. You are pumping in 2 protons into the lumen. So when these 2 protons are coming into the lumen, and in the lumen, the water splitting also happen. 2 water molecules, they give rise to 4 protons, to 4 electrons, okay, and they also evolve oxygen, okay. So when this is happening, basically you are getting abundant protons in the lumen. There is an increase in the concentration of protons in the lumen. That's why there is something called as a proton pump that is now working and leading to ATP synthesis, that we will see how it happens in the upcoming lectures, okay. Now this photolysis of water, which I told you, water splitting or oxygen evolving complex. So basically this is the complex that is there. Okay, so here water is getting splitted to oxygen, protons and electrons. These electrons, basically what is happening, so what is the need for the water to be splitted? So basically what is happening is, as soon as the PS2 or P680 is getting electron deficient because it transferred the electron, this P680, it is taking the electrons, okay, from somewhere, and that somewhere it is also taking electrons from somewhere, and that somewhere is getting the electrons by splitting of water, because when you split water you get this, if you split two water molecules you get this 4 electrons. So that is how electrons are being balanced in the entire process. So here this is the water splitting, this is for one water molecule. Okay, so 4 manganese ions are essential cofactors associated with each of the complex. Okay, apart from manganese you required this chlorine and calcium also. Okay, these are also required. Then final electron donor to PS2 if you talk about. Okay, so the tyrosine residue in the D1 protein complex, I told you CP43, CP47, D2, D1, you are having this D1. This D1, it consists of a tyrosine residue. Okay, so that is the final electron donor to the PS2. Okay, and like this is the question, that how many oxygen molecules, electrons and protons are produced when one water molecule is lysed? I have already answered the question since my first lecture, that whenever you are splitting the water, if you are splitting two water molecules, okay, you are getting 4 protons, 4 electrons, 1 molecule of oxygen. If you split one water molecule, you will get 2 protons, 2 electrons and half mole of oxygen. This is something, the stoichiometry that you should remember. Okay, so this is about the photolysis, and if we consider this process in detail, let's understand what is that something in something which I told in the previous slide. So now if you consider, basically if you are exciting this P680 with a lambda less than equal to 680 nanometers, what is happening is this is getting oxidized, it is getting electron deficient, and when it is getting electron deficient, what is happening is, since it has given its electrons, basically it requires electrons. So what is that Z that I told you? That Z is nothing but that is a tyrosine residue at the amino acid position 161. That is nothing but that is tyrosine 161. Okay, tyrosine residue at position 161, which is giving the electron to the deficient P680. Okay, this tyrosine Z is giving 2 electrons to this deficient P680, which has given the electrons further to pheophytin. Okay, and this entire thing is in the thylakoid membrane, and this is the lumen. So basically, from where is the tyrosine residue getting the electron? It is getting from the MSP, that is the complex, that is oxygen evolving complex only. Okay, or water splitting complex. So how is this MSP able to generate electrons? It is splitting two water molecules, it is releasing oxygen, it is getting some protons, 4 protons and 4 electrons. So these 4 electrons, what they will do? Basically what will happen is, as soon as it reaches to the M, okay, this is in the S0 state, it releases an electron, it becomes M plus. Then this M plus, it again releases the electron, okay, it becomes M2 plus. Then it again releases the electron, becomes M3 plus, and again releases electron, become M4 plus. And this 4 electrons, they stabilizes this and it again becomes N, and this cycle continues of the photolysis of water as well as the electron transmission from the MSP to the tyrosine Z to the P680. Okay, so this process continues. I hope the water splitting complex part is clear to everyone. Now I have to answer one of your question. Okay, but before answering your question, let's see the calculation part of it. So I told you that if you are moving this one electron in the electron transport chain, as this PQH2 is getting oxidized to PQ, It is pumping in 2 protons into the lumen side. It is pumping these in. Okay, so 2 protons are transferred into the lumen, right? And for each electron transfer in etc, 2 protons are transferred. So once you are lysing the water, you are generating 4 electrons. So for 4 electrons, you will be getting 8 protons, correct? And 4 protons you got from the photolysis of water. That when you broke water, you got 4 protons. So 4 protons are also inside the lumen. Total you get 12 protons. Total you get 12 protons. And the ATP synthase, basically that uses 3 protons. Okay, that the ATP synthase that is present, okay, what it does is that with each of the 4 protons that are there, it is able to synthesize one ATP. So if you are having 12 protons, you can synthesize 3 ATP molecules, okay, and you can also synthesize 2 molecules of NADPH2. Okay, so this is something that is happening with the help of this photolysis of water. Now, how is this NADPH2 and all this happen? That I will tell you in the next lecture of cyclic and non-cyclic photophosphorylation. Now, I hope you guys remember one important point that I have taught you in the previous lecture, that in case of photooxidation, in the second line of defense, right? In second line of defense, what you were having? In the second line of defense, you were having this scavenging molecules. Okay, you were having these scavenging molecules, like you were having the beta carotene vitamin A, then ascorbic acid vitamin C, tocopherol vitamin E, all these you were having. Apart from the scavenging molecules, there was another mechanism which was the damage to D1 protein. Now what is D1 protein of PS2, you got to know. So if the D1 protein is damaged, electron flow will be blocked and plant will not be able to do excessive photosynthesis, and the damage to the plant due to excessive light will be reduced. So that is the concept behind damaging the D1 protein, that is to block the electron flow, and plant will stop utilizing light, stop using excess light. So this is the whole thing about that D1 protein damage. Okay, so if this part is clear, then we can solve the questions. First question is given below are few statements regarding light reaction of photosynthesis. Photosystem II oxidizes water to oxygen and in the process releases protons into the lumen. We have seen this PQH2, 2PQ, 2 protons released into the lumen for each electron transfer. Okay, so this is correct. Photosystem II oxidizes water and in the process it put into stroma. No, that is wrong. So this everything happens in the lumen, the proton release. In the stroma, nothing is released. Okay. So in lumen there is a high concentration of protons, in the stroma there is low concentration of protons. ATP synthase, this is wrong. ATP synthase produces ATP while protons go back through it from lumen to stroma. Absolutely correct, guys. So here, if this is your stroma, this is your lumen, there is something you called as this ATP synthase, and there is a high concentration of proton, there is a low concentration of proton, there is a movement of the proton from the lumen to the stroma. In this process, due to the high energy of the protons, it moves like, because of the wave nature and all this energy part that is involved, there is a conformation change on the head of the F0, F1 particles of the ATP synthase, and because of that change, we are able to couple ADP with PI and generate ATP. So this part is correct because of the movement from lumen to stroma. What are the factors which contribute to increase in proton concentration in the lumen? One is the photolysis, water splitting. Another one, this PQH2 to PQ. Okay, so the correct answer is A and C. D is wrong because another direction. Stroma to lumen, it doesn't happen. So this is wrong. Okay, very good everyone, if you are able to solve it. Now, names of some genes in column X and their involvement in photosystem 1 or photosystem 2 is provided in column Y. Which one of the following combination represent correct matches between column X and column Y? I told you the trick with PS or any sort of gene, any abbreviation, let it be X. If there is A written, A means what? A means PS1. If there is B written, okay, if there is B written, B means PS2. So wherever you find A after the prefix, that belongs to PS1. That means B and D, they belongs to PS1, and wherever you get B after the prefix, that belongs to PS2. So that means B and D belongs to PS1, B and D belongs to PS1, that is second, and A and C belongs to A and C belongs to PS2, that is first. Okay, so this is the correct answer, 1. Okay, so that is how you can solve all these kinds of questions. One more question on the inhibitor. The herbicide dichlorophenyl dimethylurea DCMU is an inhibitor of schemate pathway for biosynthesis of aromatic amino acids. Does I ever took the name of schemate pathway? No, this is not. Electron transport from P680 to P700, absolutely correct. It blocks the transfer from this QA to QB in the PS2, and if this transfer is blocked, then electrons will not be transmitted to the PS1. So that is correct. Branch in amino acid pathway, no. Electron transfer from P700 to ferredoxin, no, no, no. That is something that is to done with the another inhibitor, okay, which we have studied. So that is also wrong. Yeah. So these were some of the questions. Thanks a lot everyone for joining with today's lecture. In the next lecture, we will be dealing with cyclic and non-cyclic photophosphorylation. I hope this lecture is clear to you, everything that we have studied. Okay, thanks for joining.